Hello, my name's Austin Worth, and I'm giving the third and final infectious disease lecture for the MRC PCH Part 1 past tests lecture series. This lecture is entitled HIV, TB and Tropical Infections. The learning objectives for this talk are to try to give an overview of HIV, TB and malaria, highlighting some of the topics I think are more likely to come up, and provide a summary of new drugs and new diagnostics that are being used in the management of these conditions. I then hope to introduce some slightly more unusual topics in tropical infections, which is a bit of a vast area, and we have to take a bit of a gamble and just pick a few to cover them. But hopefully it should highlight some of the some patterns that will be relevant for other conditions as well. I would just point out that there's quite a lot of information in this talk, and I am going to have to run through it pretty quickly, so you might find that there's a bit of information overload at points, but hopefully most of the information is set out fairly well, so you can come back and have a look at it in your own time. So question 1. HIV transmission from mother to child can be decreased by which of the following? A caesarean section. B, treating the child after birth for 6 weeks with antiretrovirals. C, treating the mother with antiretrovirals in pregnancy. D, not breastfeeding. E, treating the infant with prophylactic antibiotics. What do you think? Right, the answers are, they're all true except for the last one. So, vertical transmission of HIV is a pretty hot topic generically, because it's somewhere where we can intervene and actually do something. So from the mother's perspective, the key fact here is that maternal viral load correlates with transmission rate. So therefore, we need to monitor viral load throughout pregnancy, and at the moment, the policy is to be doing that at 3 months, 36 weeks, and at delivery. At present, there's no evidence for teratogenicity with highly active antiretroviral therapy, or HAART. However, HAART is associated with prematurity. So the national guidelines now recommend that if the mother's on HAART already, then not to change anything. If the viral load is greater than 10,000 copies per mil, the mother should be started on HAART at somewhere between 20 and 28 weeks gestation. If it's less than 10,000 copies per mil, she should receive AZT monotherapy. In terms of the delivery, planned caesarean sections for mothers on AZT monotherapy or with a viral load greater than 50. If the viral load's less than 50, then a vaginal delivery should be safe and should be offered, even if the mother is on HAART and the viral load is undetectable. However, there are some caveats, and invasive monitoring should be avoided. If the mother goes into labour with a viral load greater than 50, then intravenous AZT or zidovudine should be commenced intrapartum. For the baby should receive 4 weeks of AZT monotherapy unless the mother was viremic and untreated, in which case the baby should receive 4 weeks of highly active antiretroviral therapy, i.e. 3 drugs. And the baby should be exclusively bottle-fed. So assessing immunity of children with HIV is a little bit different to adults, because children have much higher CD4 counts than adults do. So it's more appropriate to use percentage CD4 counts. And the levels of immunosuppression based on percentage CD4 count are set out here. So greater than 25% is no suppression, 15 to 24 is moderate, and less than 15 is severe suppression. As a child becomes more immunosuppressed, there is a number of common opportunistic infections that they're susceptible to. And the ones stated down here are the AIDS-defining opportunistic infections. So extensive candidiasis, so oropharyngeal rather than just in the mouth. cryptosporosis in the stool, disseminated histoplasmosis, disseminated CMV, atypical mycobacterial infection, pneumocystis jirovecii, and persistent or disseminated HSV, or toxoplasmosis in the brain. Lymphoid interstitial pneumonitis is an interesting phenomena that children get, that we don't fully understand why some children get it and why others don't. About 40% of children with vertically acquired HIV appear to go on to develop this, and it is actually a good prognostic sign. It's associated with lymphoproliferation, so there's what appears to be a lymphocytic infiltration on the chest X-ray with interstitial shadowing. There's often parotiditis. There's often a very high IgG level, greater than 50 units per mil. And here you can see chest X-ray with a sort of characteristic appearance of LIP involving sort of symmetrical lung fields, really quite hazy. Okay, with the advent of new antiretroviral therapy, it's important to have at least a basic understanding of the HIV life cycle to know where all the drugs fit in and how they act together. This slide shows or describes the steps of the HIV life cycle. Text that's written in red refer to proteins or enzymes or glycopeptides that are virally derived, whereas words in blue refer to host protein or mechanism that the virus has hijacked to be able to maintain its life cycle. So when an HIV virion is infecting a cell, the first thing it has to do is it has to attach. And to do this, it uses the HIV protein GP, or glycoprotein 120, which binds to host CD4. The next stage is fusion, and fusion of the outer surface of the HIV virus and the injection of the nucleocapsid into the cytoplasm. And for fusion, CCR5 or CXR4 on the host cell are essential for activating the viral GP41. CCR5 is found on macrophages, dendritic cells, and CD4 cells, and this tends to be recognized by HIV variants, which are macrophage trophic, the so-called R5 variants. This tends to occur earlier on during the HIV infection. CXCR4, however, is found on activated T cells, and this tends to attract viruses that are lymphotrophic, the so-called X4 variants, and this tends to be an indicator of late infection. Once the viral proteins and enzymes have been released into the cytosol, the next stage of the virus is to reverse transcribe the double-stranded RNA, which is retroviral genome, to double-stranded DNA. Once this has been performed, then an enzyme called integrase hijacks host DNA repair proteins and instigates the transport of the viral DNA through the nucleopores into the nucleus and to be integrated into the host genome. Once this occurs, viral DNA integrated into the host genome will hijack the host protein production machinery to generate viral proteins. These will then be trafficked to the cell surface, where they'll be packaged and arranged on the cell surface before the virus is budded and finally released out and exocytosed. Finally, once the mature virus has been budded, the virus proteins themselves are actually matured by an enzyme called protease, which is a viral enzyme, that's essential for just the very final step of viral maturation. So this life cycle is illustrated in this cartoon, and it also demonstrates the sites of action of many of the antiretroviral drugs. We've had for quite some time the original drugs, the NRTIs, or the nucleoside reverse transcriptase inhibitors, and the second line of drugs, the non-nucleoside reverse transcriptase inhibitors, act at the point in which the viral double-stranded RNA is converted to double-stranded DNA. The third generation of drugs that came along were the protease inhibitors that acted by inhibiting the very final step just as the virus or just after the virus has budded, preventing the HIV virus from attaining full levels of maturity. So now we have a new class of drugs called the fusion inhibitors, an example of which is T20, which acts as a CCR5 antagonist and therefore blocks the co-receptor that's required by GP41 for the virus to fuse with the cell and actually enter the cell cytoplasm. So this slide summarises the antiretroviral drugs that are currently in use for children, or soon will be in use for children in this country. I've just put on the bottom of it the side effects just for reference as well. So the nucleoside reverse transcriptase inhibitors have problems with lactic acidosis, gastrointestinal upset, and one of the more major side effects that limits their use is lipodystrophy, which is the redistribution of fat reserves around the body. Both the NNRTIs and the protease inhibitors interfere with the P450 breakdown system in the liver. However, NNRTIs are inducers, whereas the protease inhibitors are inhibitors. All the drugs have the potential to give rashes, nausea, and vomiting, and generally they remain fairly unpleasant drugs to take, despite the fact that the actual formulation of them has improved very significantly, and now many older teenagers or adults can get away with a far smaller number of drugs than they had to 5 and 10 years ago. So question 2. Pulmonary TB in children is A, usually more infectious than in adults, B, excluded by 2 negative sputum smears, 3, excluded by a negative MON2 test, D, treated with rifampicin and isoniazid alone, E, usually primary disease. Okay, and the answers to this are the first 4 are false, and the final one is true. Any question that has 2 stems that have excluded by, you know you're onto a fair old chance that they're going to be false and negative. So on to TB. The causative organism is Mycobacterium tuberculosis, which is a non-motile bacillus, an obligate anaerobe, which has a cell wall that is fast, i.e. unaffected by both alcohol and acid and most standard stains. And it only staining up a red colour with the Zeal-Nielsen stain. Hence the phrase, the description, acid-fast bacillus. TB is spread by droplet spread, and the smaller the droplets, the more infective it is. It can also be transmitted transplacentally and in breast milk, and other Mycobacterium such have been transmitted by cow's milk. There are many other species of Mycobacterium, many of which do cause disease in children as well. And these are termed atypical Mycobacterium. Probably the most important is Mycobacterium avium and Mycobacterium bovis. So to begin with, I'm going to summarise the natural history of TB with particular respect to children. So the first encounter is exposure.